Good morning, good afternoon. My name is Dr. Mohammed Ezzi. I'm a medical oncologist and a lecturer at the University of Nairobi. I'm now going to discuss about the Boston study. The Boston study was a phase 3 study that looked at the role of Selinexor in patients who had received prior treatment for multiple myeloma. This study was presented by Melchius A. Dimopoulos. Patients with multiple myeloma continue to relapse despite all of the new therapies which indicate a need for therapies with new mechanisms of action. Selinexor is a first-in-class selective inhibitor of the nuclear export targeting the nuclear transport protein exportin-1. By doing so, it interferes with the transport of proto-oncogenesis and apoptosis-related proteins between the cytoplasm and the nucleus. This drug is effective in combination with dexamethasone in relapsed multiple myeloma, and based on the data from single-arm trials, the FDA approved Selinexor in combination with dexamethasone for patients who have had more than 3 previous therapies and have been exposed to more than 2 immunomodulatory drugs and more than 2 proteasome inhibitors, and have also received an anti-CD38 monoclonal antibody. The Boston trial was designed to explore the role of selinexor in patients who had received 1 to 3 previous therapies. Patients with relapsed multiple myeloma who had 1 to 3 previous lines of therapy and measurable disease were randomized to receive a standard dose of Bortezomib and Dexamethasone at a schedule of twice-weekly injections. The schedule of twice-weekly Bortezomib in a 3-week cycle for a total of 8 cycles. This was followed by maintenance therapy with a weekly injection of bortezomib dexamethasone in a 5-week cycle. In the experimental arm, patients received selinexor in combination with bortezomib and dexamethasone. However, bortezomib was administered weekly in the experimental arm from the beginning in 5-week cycles. The primary endpoint was progression-free survival. 402 patients were enrolled in the study, with 195 participants in the selinexor arm and 207 patients in the bortezomib dexamethasone arm. 80% of the enrolled patients discontinued treatment in both arms, 50% in the VD arm and 34% in the Selinexor arm, and they discontinued due to disease progression. A third of the patients died in the VD arm, while a quarter of the patients died in the Selinexor arm. The patients were well balanced between the two arms. About half of the patients in the study cohort had high-risk cytogenetics, and 80% had had previous 2 proteasome inhibitors. The primary endpoint was made because the addition of selinexor to dexamethasone and bortezomib significantly improved the progression-free survival by 4.5 months. The hazard ratio of 0.7, that implies a 30% reduced risk of progression, and a p-value of 0.0075. Subgroup progression-free survival analysis showed that selinexor added benefits in all patient groups, particularly in patients with deletion 17p, which signifies a high-risk cytogenetics, and also in patients who had previously not been exposed to a proteasome inhibitor. The addition of Selinexor improved the overall response rate from 62% to 76%, p-value of 0.0012, compared with dexamethasone and bortezomib alone. This improvement was seen in all patient subgroups, including patients 65 years of age and older with a p-value of 0.0243, patients with high-risk cytogenetics, p-value of 0.00008, and patients with previous bortezomib treatment with a p-value of 0.0005. And it was independent of the underlying renal function, the p-value of 0.0055. The duration of response during this among the 72% of patients who responded with Selinexor was 20.3 months, while it was only 13 months for patients in the VD arm. The time to next therapy was also significantly longer at 16.1 months for Selinexor plus bortezomib and dexamethasone versus 10.8 months for bortezomib and dexamethasone with a hazard ratio of 0.6 and a p-value of 0.0012. In the interim analysis of the median overall survival, in the Selinexor, the median overall survival had not been reached, while it was 25 months in the bortezomib dexamethasone arm. Thrombocytopenia of any grade was 60% with Selinexor plus VD versus 27% with VD only. 35 patients in the Selinexor arm required a thrombopoietin receptor agonist compared with 2 patients in the VD arm. Several specific toxicities, including nausea 50%, fatigue 42%, and anorexia 35%, were the most common adverse effects of any grade seen in patients treated with Selinexor, and were more common in these patients than in those treated with VD. A significant burden of a GI adverse effects was seen with the addition of Selinexor. Based on some of the results from this study, as well as others, prophylactic use of antiemetics can help alleviate this toxicity. Weight loss and asthenia were more strongly associated with Selinexor, which was expected because of the GI toxicity of this drug. Peripheral neuropathy was the most common cause of discontinuation of treatment in these patients, but was significantly less in patients who in the Selinexor arm than in patients in the VD arm only, 32.3 versus 47.1% respectively, with a p-value of 0.010. This is probably due to the fact that patients in the Selinexor plus the VD arm were receiving bortezomib weekly compared with the twice-weekly dosing of bortezomib in the VD arm. The Boston trial confirms the results of the phase 2 studies in terms of efficacy of Selinexor, but it also highlights its significant toxicity, which requires careful management with antiemetics and appetite stimulants. A different class of drugs for the treatment of multiple myeloma will always be welcome. Selinexor is convenient because it is taken orally once a week. This allows us to create more oral combinations, which is especially important for the treatment of all the patients with multiple myeloma. Other combinations now being investigated in phase 2 trials include Selinexor combined with carfilzomib or daratumumab. Phase 1 and 2 trial data showed that selinexor has similar overall response rate when combined with daratumumab or carfilzomib. Thank you very much.